Good morning, everyone. My name is Kenny Wu, and I'm the program coordinator here at Walker LCTSE. Thank you, everyone, for joining us today on our COVID-19 and the Community, Part 2. Today, we're joined by Jonathan Berardi, who is a research nurse practitioner with the Cornell Clinical Trials Unit at Walker Medicine. The presentation will be done in English and Spanish. If you want to ask any questions or want to talk to a presenter, there's a chat function at the bottom of the screen. You can type in a question and the presenters will answer during the Q&A session of the presentation. At the end, we will have a short poll about today's presentation. Please fill it out if possible. Muy buenos días, mi nombre es Kenny Wu y yo soy el coordinador de programa en Walker CTSC. Muchas gracias a todos ustedes por atender a nuestra presentación el día de hoy. El día de hoy vamos a tener una presentación sobre el COVID-19 y la comunidad, parte 2. Hoy nos acompaña Jonathan Berardi, quien es un enfermero practicante e investigador de la unidad de ensayos clínicos en Walker Medicine. La presentación va a ser en inglés y español. Si tienen preguntas, en la parte de abajo hay una sección donde nos pueden enviar un chat o un mensaje. Al final vamos a tener una encuesta sobre la presentación. Por favor, llénenla si pueden.
Thank you for that introduction, Kenny, and welcome everyone to part 2 of the COVID-19 community series. So today we're going to be focusing on COVID vaccines as well as vaccines in general to gain a deeper understanding of vaccine history, the technology, and how we've been able to use or implement new technologies in the setting of COVID-19. So we're going to discuss the historical development of the vaccines that we have already, understand how those vaccines generate immunity, identify different types of vaccines, and discuss and understand the current COVID vaccines that are either in use through the emergency use authorizations or in development.
El día de hoy vamos a hablar, los objetivos del día de hoy son comprender el desarrollo histórico de la vacuna, comprender cómo las vacunas generan inmunidad, identificar los diferentes tipos de vacunas y vamos a discutir y comprender las vacunas de COVID-19 actuales en uso y en desarrollo.
So we'll start with a brief history of vaccines.
Vamos a comenzar con una breve historia de COVID-19.
So initial vaccine development was a very slow process, but as science advanced, we were able to come up with new vaccines at a faster scale. And the table here just shows that the very first vaccine that was developed was smallpox in the late 1700s. And then it took another 100 years before we had another four in total. And then as you see in the 1900s, the number of vaccines that were developed steadily increased. And we saw this large increase in the 1950s to 1990s.
El desarrollo de la vacuna. El desarrollo inicial de la vacuna fueron un proceso lento, pero aumentó rápidamente a medida que la ciencia y la tecnología se hicieron más avanzadas. Por ejemplo, de 1700 a 1800, el número de vacunas desarrolladas fue una. De 1800 a 1900 fueron 4. De 1900 a 1940 fueron 7. Y así incrementaron, ¿no? Hasta en 2000 a 2019, 10.
So to understand vaccines, I want to go back to the very first vaccines that were being used. And before we had what we now call vaccines, we had a process called inoculation. And that word comes from Latin meaning to graft. So we would inoculate individuals against an infection. The inoculation process was used in the prevention of smallpox. And smallpox was also called variola, and it still retains that name, but we now commonly call it smallpox. The word inoculation became interchangeable with this word variolation, which comes from that variola, smallpox. What they would do is they would take a small amount of infectious material, so somebody who had smallpox, and they would place it into a cut or a scrape made in the skin of somebody who was healthy. And what they observed was that that tiny exposure allowed somebody to develop immunity against smallpox. So communities were able to be preventatively treated. This technique was used as early as the year 1000 and developed independently across the world. So a lot of different cultures and civilizations had the same observation at roughly the same time, even though they were geographically isolated.
Para comprender qué son las vacunas, vamos a hablar sobre las primeras vacunas. Antes de tener las vacunas que conocemos el día de hoy, se usaba una técnica llamada inoculación para conferir inmunidad. De latín inoculare, injertar. Se utilizó contra la viruela, antes llamada variola. La palabra inoculación se volvió intercambiable con variolación. Ah, ¿Y cómo se hacía? Se colocaba una pequeña cantidad de material infeccioso en un corte o rasguño hecho en la piel. Esta técnica se utilizó desde el siglo XI y fue desarrollada de forma independiente en todo el mundo.
So in between the year 1000 and the early 1700s, this inoculation was a commonly known procedure. But in the 1700s, it became common knowledge that individuals infected with cowpox became immune to smallpox. Both are members of the same virus family, Poxviridae, but cowpox causes a much more mild infection compared to smallpox. And this connection was predominantly seen with milkmaids. So a physician scientist by the name of Edward Jenner conducted an investigation into that connection. What he was able to discover or identify was that an injection of the smallpox or the cowpox pustule or pus led to immunity from smallpox. He documented his results and he published them, although they weren't initially accepted for publishment, but they were the first published records of vaccines. And the reason why is because a new word was coined. That's where we first saw the word vaccination, which comes from the Latin word for cow. And that's where inoculation began that transition toward vaccination.
Inoculación y vacunación. A los principios del siglo XVIII, se hizo de conocimiento común que las personas infectadas con viruela vacuna se volvían inmunes a la viruela. La viruela vacuna es una vacuna causa una infección más leve en comparación con la viruela. Ambos son miembros de la familia Poxviridae y es predominantemente visto con la lechera. El doctor Edward Jenner en 1796 realizó una investigación sobre esta conexión con éxito y una nueva palabra acuñada fue vacunación, del latín vaca.
So what does a vaccine do? A vaccine is designed to work by triggering or training our immune response. We use a vaccine to deliver what's called an antigen. And an antigen is any piece of material. It could be an infectious material or sometimes even an allergen, although an allergen is typically something more environmental, that is recognized as foreign by the immune system. Once it's recognized as foreign, the immune system learns how to fight that off and defend itself from another infection in the future. So our immune system learns how to fight the infection, and we can reduce the risk of more serious disease. And because of this learned process, the next time we're exposed, our defenses can begin working much faster than if we were to acquire the infection naturally.
La respuesta inmune. Las vacunas actúan entrenando nuestra respuesta inmunológica para responder a una infección. Las vacunas liberan un antígeno que se reconoce como sustancia extraña y nuestro sistema inmunológico aprende a cómo combatir una infección, reduciendo el riesgo de enfermedades más graves. El sistema inmunológico puede comenzar a combatir la infección mucho antes, después de la exposición.
So this slide is a slide that was used in part 1 of this series, and it's a great image that shows how a vaccine works. It allows our body to recognize an infection, sound the alarm, and then the fighter cells go into action. The SARS-CoV-2 vaccines, the current ones, they cannot cause COVID-19. And we'll talk about why later, but they cannot cause COVID-19.
Esta imagen la usamos en la primera parte de la presentación y lo que aquí es lo que en esta imagen podemos ver cómo una vacuna enseña al cuerpo a reconocer y luchar contra los invasores. Y una de las preguntas que nos hacen es, ¿pueden las vacunas causar la infección de SARS-CoV-2 o causar la enfermedad por COVID-19? Y la respuesta es no, las vacunas que están que se están probando están hechas de piezas sintéticas fabricadas en el laboratorio, copiadas de SARS-CoV-2 y no de virus completo. Por lo tanto, las vacunas no pueden causar infección ni causarte la enfermedad de COVID-19.
The ultimate goal of vaccination is to generate what's called herd immunity. And I'm sure that that's a phrase that you may have heard already in the news and the media. The goal of herd immunity is to prevent the spread of virus and also to insulate those among us who are more at risk or more vulnerable from coming into contact with that infection. So if fewer people become ill or get sick, then it becomes harder for the virus to spread. And that's what we see in this image. So off to the side, we have the second image shows that some of the population has been vaccinated, but we still have a fairly large infection rate. Whereas if most of the population is vaccinated, then those limited outbreaks don't cause significant infection. And we are able to prevent individuals who can't be vaccinated or who are at a higher risk of severe disease from ever coming in contact with it. So for example, children, especially very young children, immunocompromised individuals, and we're breaking the chain of that infection.
El objetivo final de la vacunación es generar inmunidad colectiva. Si menos personas pueden enfermarse, el virus se vuelve más difícil de propagar. En este momento todavía necesitamos máscaras porque no sabemos si las vacunas pueden prevenir la propagación del COVID-19. En la imagen, en la primera imagen, nadie está inmunizado, la enfermedad es contagiosa, se propaga por la población. En la segunda imagen, parte de la población se vacuna, la enfermedad contagiosa se propaga a través de parte de la población. Y en la tercera imagen, la mayor parte de la población se vacuna y la propagación de enfermedades contagiosas está contenida.
So before I move on to the different vaccine platforms, I want to take a pause. Does anybody have any questions about vaccine history and development?
Yes, I have a quick question. In terms of, does history teach us anything about the efficacy of smaller doses of vaccination for those who have been already infected versus a standard dose for those who have not been infected? I'm trying to see whether or not history informs us if there's any learning, any pedagogy from the past as it relates to the new emerging theory, for example, that those who have had the virus only need one dose versus two. I'm not sure whether or not, if that is the case, it would be great news in as much as it would save and make available that many more doses, or whether or not history teaches us another cautionary. Thank you.
Sure, so that's a great question. And with the history of vaccines, we don't have a lot of information that would suggest active infection leading to a decreased need for immunization after the fact. However, we are also using very new or relatively new types of vaccine technology that we're going to be discussing in just a little while that may obviate the need for that second dose because of the way it produces that immune response. So while we don't have anything per se from history, we are, in a sense, making history now because we are investigating if this is a true finding and if, in fact, somebody that has had COVID may not need a second dose.
Thank you so much. That's a triple. Thank you. Thank you.
Okay, so discussing the platforms for the vaccines.
Vamos a hablar sobre las plataformas de vacuna.
So when we say a vaccine platform, what we're talking about is the delivery system that we use to promote or produce that immunity. The current vaccines that we have fall into one of four groups. So we have our Live-Attenuated Vaccines, Inactivated Vaccines, Subunit Vaccines, and Toxoid Vaccines. And we'll discuss those in depth as we move forward.
¿Qué son las plataformas de vacuna? Una plataforma de vacuna es el tipo de sistema de administración que se utiliza para promover la inmunidad. Las vacunas actuales se dividen en uno de 4 grupos. Estas son vacunas vivas atenuadas, vacunas inactivadas, vacunas de subunidades y vacunas con toxoides.
The first in that list were the Live-Attenuated Vaccines. So these vaccines use a weakened form of the virus to generate an immune response. And we call that weakened form an attenuated form. And the way that we weaken it is by making it less virulent, but the virus still stays active enough that we have an immune response. Vaccines of this type tend to produce very long-lasting immunity. So boosters are not often needed after a series has been completed. Although these vaccines are a weakened or attenuated form of the infection, not everyone is an ideal candidate to receive this type of vaccine. Individuals who have any immunosuppression or who may be pregnant are not recommended to receive live vaccines. This is why it's important that we advise and keep to our vaccine schedules as best as possible to mitigate any future risk of that.
Las vacunas vivas atenuadas. Estas vacunas utilizan una forma debilitada de un virus para generar una respuesta inmunitaria. Describimos esta forma debilitada como vacuna atenuada. Las vacunas de este tipo producen una inmunidad duradera. Los impulsores no se necesitan a menudo después de completar la serie de vacunas. Y aunque debilitado, no todo el mundo puede recibir esta clase de vacunas, como por ejemplo las personas que tienen inmunosupresión y las personas que están embarazadas.
Some examples of live attenuated vaccines include measles, mumps, rubella, varicella, which causes chickenpox, rotavirus, smallpox, the polio oral vaccine, and some forms of the flu shots. And I always like to note that the measles, mumps, rubella, and varicella are often combined into one shot, which is called the MMRV. Before we had the varicella immunization or vaccination, we had the MMR series. So it's nice when we can combine some of these into one injection rather than 3 or 4.
Y aquí hay ejemplos de las vacunas vivas atenuadas, como por ejemplo la sarampión, paperas, rubiola y la varicela, rotavirus, la viruela, polio y algunas formas de vacunas contra la gripe. Las primeras 4, que son la sarampión, paperas, rubiola y la varicela, a menudo estos se combinan en una sola inyección, la MMRV.
The next group of vaccines on the list are the inactivated vaccines. So this type of vaccine uses a killed infectious agent to produce an immune response. So unlike the live attenuated, where we use a weakened form, with the inactivated vaccines, we actually just use the dead infectious agent. These can either be killed using heat or specific chemicals to neutralize them and make them inactive. This prevents, because we're inactivating them in this way, it prevents the virus or the agent from being able to cause an active infection. However, because we don't have that same virulence, these types of vaccines produce long-lasting immunity, but do require boosters periodically throughout an individual's lifetime.
Las vacunas inactivadas. Este tipo de vacuna utiliza una forma muerta del agente infeccioso para reducir una respuesta inmunitaria. El agente infeccioso, como las bacterias, virus, etc., se eliminan mediante calor o productos químicos específicos. Esto evita que pueda causar una infección después de la inyección. Este tipo de vacunas producen una inmunidad duradera, aunque periódicamente se requieren esfuerzos para mantener esta inmunidad.
Some examples of inactivated vaccines include the annual flu shot, and that's the more common type. Hepatitis A, which is generally recommended depending on workplace or exposure risk, and the injectable form of the polio vaccine. The injectable polio vaccine is the only type of polio vaccine administered in the US, but we do have the injectable as well as the oral form. And the oral form is a live-attenuated that's administered in other parts of the world.
Ejemplos de vacunas inactivadas: la vacuna anual contra la gripe, la vacuna de hepatitis A y la vacuna contra polio, que es la vacuna inyectable. Este es el único tipo de vacuna contra polio que se administra en los Estados Unidos, pero también hay vacunas para el polio, contra el polio, que son vacunas orales, y estas vacunas son vacunas vivas atenuadas.
Third on our list are the subunit vaccines, and this category of vaccine uses parts of pathogens, so subunits, smaller parts of the whole, rather than the whole cell or virion. Because we're administering smaller parts, we use something called an adjuvant, which enhances the immune response. Again, because these are smaller parts and because we're using the adjuvants, these types of vaccines tend to require boosters throughout an individual's lifetime. Included in the subunit vaccine category are what we call polysaccharide vaccines, conjugate vaccines, and recombinant vaccines. Polysaccharide vaccines use the sugar coating that's found on the outside of bacterial cells to stimulate the immune response. So you don't become infected with the bacteria, but you become infected or immunized with a part of the bacteria to train the immune system to recognize and stimulate that response. The conjugate vaccines combine stronger antigens with weaker antigens. And the goal of that is to produce an immune response to the weaker part. So some of these subunits are so small or so fragile that they alone would not be able to produce enough of an immune response. But when we're training our immune system, it's like we're training for a race. We want to go out there and we want to train hard so that we can win the race. And our immune system wants to do the same thing. And to train, we need to have a stronger initial presentation. So that's where these conjugate vaccines are used. The recombinant vaccines use a process of taking the genetic material from a disease and placing it into a cell in a lab. The proteins that are then produced are used for the vaccines. So we're not using any active infectious piece, but what we're doing is we're taking a harmless bacteria and something that can produce an infection like a virus. We're taking the genetic material, we're combining them together to get a protein at the end that our immune system can use to train.
Vacunas de subunidades. La categoría de subunidades de vacunas utiliza partes de un patógeno en lugar de una célula completa o virión o un virus completo. Los ayudantes se utilizan para mejorar la respuesta inmunitaria y los refuerzos se requieren con mayor frecuencia porque solo se utilizan partes. Este grupo incluye lo siguiente: las vacunas de polisacáridos utilizan azúcares que se encuentran en el exterior de las células bacterianas para estimar la respuesta inmunitaria. Las vacunas conjugadas combinan antígenos fuertes con antígenos más débiles para producir una respuesta fuerte a la parte más débil. Y las vacunas recombinantes utilizan un proceso de inserción de material genético de una enfermedad en las células de un laboratorio. Las proteínas producidas se utilizan para la vacuna.
Here are some examples of subunit vaccines that are administered. So we have the pneumonia meningitis shots, and that is a type of polysaccharide vaccine. So those are two vaccinations that can protect against bacterial infection. We also have the conjugate vaccine grouping. So we have the shingles shot, the HPV shot or human papillomavirus, another type of pneumonia shots, the PCV13, and typically you need to receive one of each. Haemophilus influenzae type B, hepatitis B, meningitis, and acellular pertussis are considered recombinant vaccines.
Aquí están los ejemplos de vacunas de subunidades. Por ejemplo, vacunas contra la meningitis, la neumonía, PPS V23, son vacunas de polisacáridos. La vacuna contra herpes, virus de papiloma humano, neumonía y Haemophilus influenza tipo B, que son vacunas conjugadas. Y en vacunas recombinantes tenemos la hepatitis B, meningitis y la tosferina acelular.
And before I move to the next slide, I did see in the chat a myth unmasked. The vaccine does not inject COVID-19 in the body. So we're actually going to be talking about that in just a few slides. So great question, and keep me on my toes. The last type of vaccine on the list are the toxoid vaccines. So these vaccines use toxoids. Toxoids are produced by infections. They're proteins that are secreted. And the reason why we use toxoid vaccines is because the pathogens themselves are so powerful that we can't safely use them in other vaccine methods or with other vaccine platforms. A small amount of the infection could prove lethal. So instead, we use the toxic proteins that are produced by those infections to generate an immune response so that our immune system can begin fighting off the infection based on recognition of the toxoid.
Hay un comentario en el chat que dice que las vacunas no inyectan el COVID-19 en el cuerpo. Es una percepción errónea generalizada. Ahora vamos a hablar sobre las vacunas con toxoides. Algunos patógenos son tan poderosos que no podemos utilizarlos de forma segura en una vacuna utilizando otros métodos. En su lugar, usamos proteínas tóxicas producidas por estas bacterias infecciosas para entrenar el sistema inmunológico. Estas proteínas las llamamos toxoides.
Examples of toxoid vaccines are diphtheria, which is an infection that causes destruction of healthy respiratory tissue. Tetanus, which is caused by a bacterium called Clostridium tetani, and it can cause lockjaw. Pertussis, more commonly called whooping cough because of the distinct sound of its cough. And these three toxoid vaccines are often administered as combination shots. So we have the DT or TD, which is a combination of diphtheria and tetanus only. We often administer this after somebody has experienced a laceration or gets cut with a rusty piece of metal, for example, or any soil contaminated sharp object. We have the Tdap, which is recommended as a booster every 10 years, the tetanus, diphtheria, and acellular pertussis. So we use that to boost for the whooping cough as well. And we recommend that people get a booster of this before newborns come into the home. And the final combination of that is the DTaP, which is the pediatric version of the Tdap. So there are some differences. There are some age restrictions. And for children under the age of 7, they can only receive a DTaP.
Ejemplos de vacunas con toxoides: difteria, inyección que destruye el tejido respiratorio sano. Vacunas contra tétanos causados por Clostridium tetani y causa trismo. La tosferina, llamada tosferina debido al sonido distintivo de gritos de la tos. Y a menudo se administran inyecciones combinadas, como por ejemplo la DTOTD, que son la difteria y tétanos solamente. Tdap, que son tétanos, difteria y la tosferina celular. Y DTaP, inmunización pediátrica para los 3, pero es para niños menores de 7 años.
Before we move on to the COVID vaccines, any questions about the current vaccine platforms that we use?
Yes, there is a question. I'll say in English and then in Spanish. Are there any treatment options besides getting vaccinated? ¿Existen otras opciones de tratamiento como refuerzo, además o aparte de vacunarse?
For COVID-19, there are treatments that are either in use in specific situations or in clinical trials right now. The treatments are typically monoclonal antibodies, which are available through the emergency use authorization. There are 2 EUAs that were issued. One is for a monoclonal antibody called bamlanivimab, and another is for a compounded combination that was issued to Regeneron. Those are the only treatment options we have right now, and they are limited, but we do have more that are in clinical trials at this time. We also have a medication called remdesivir, which is an antiviral medication that we can give if somebody is in the hospital.
Por el momento, los tratamientos contra COVID-19 son limitados. Los tratamientos que tenemos en el momento, por ejemplo, tratamientos de anticuerpos monoclonales por uso de emergencia, y también se le da remdesivir a las personas que están hospitalizadas. And I think that's it in terms of questions.
Okay, so now we'll talk about the COVID-19 vaccines.
Ahora vamos a hablar sobre las vacunas para COVID-19.
Right now, there are 2 vaccines that have received emergency use authorization from the FDA, and there are additional vaccines that are being evaluated in clinical trials. The New York Times maintains a COVID vaccine tracker, which is actually very interesting to look at. And I placed the graphic here. This is information as of February 18th. So you can see that there are a different number of vaccines that are in different phases of investigation, where phase 1 is very limited. Phase 2 is where we begin an expanded investigation into the general population. Phase 3, we enroll even more participants or individuals. And then companies may seek approval or authorization. Just as a comparison, the last time that I had a graphic for this vaccine tracker, there were the same number of vaccines in phase 1 testing. Phase 2 testing has actually increased from 24 to 28. The phase 3 testing has remained the same. The number of limited vaccines was 8, whereas here we have 6, and we have 4 total approved, whereas again 2 weeks ago we only had 2 approved. And the number that have been abandoned in clinical trials remains the same at 4. So it's interesting to see how the numbers are changing as the weeks go on.
Oh, I actually have the older the slide that we had last time here for the comparison. Yeah, just for the comparison. Y sobre las vacunas, hay 2 vacunas que han recibido autorización de uso de emergencia de la FDA, y se están evaluando vacunas adicionales en ensayos clínicos. Estas vacunas son la de Pfizer y la de Moderna. Y en comparación, can you go back to the slide from before? Yeah. Aquí lo que tenemos, hay 28 vacunas en fase 2. Estas vacunas ensayo de seguridad ampliado. Hay 20 vacunas en fase 3, vacunas en pruebas de eficacia o a gran escala. Y hay 6 vacunas que están en uso temprano o uso limitado. 4 vacunas que han sido aprobadas para uso completo y 4 vacunas abandonadas después de los ensayos.
And what I want to point out about this graphic as well is that although 4 vaccines are approved for full use, that doesn't necessarily mean that they are approved for full use in the United States.
Aunque hay 4 vacunas que son aprobadas para uso completo, eso no significa que estas 4 vacunas han sido aprobadas para uso completo en los Estados Unidos.
So here are some examples of the vaccines that are in trials, as well as a little description of what or where they might be. So in addition to the Pfizer and Moderna trials, which are ongoing, we have the Oxford AstraZeneca vaccine trial. They received emergency use authorization in the United Kingdom, and there's an ongoing trial here in the United States. There's also the Janssen trial, which is being led by Johnson & Johnson. They have actually applied for emergency use authorization. They applied at the beginning of this month, and we're hoping to hear some news by the end of this week as to whether or not it's approved for an EUA. And what's different about the Janssen vaccine compared to the rest is that it only requires 1 shot instead of 2. We also have Novavax trials, and Sanofi is going to be engaging in clinical trials as well.
Aquí están las vacunas de ensayos. La de Oxford AstraZeneca está en uso de emergencia en el Reino Unido. Y hay un ensayo en curso en los Estados Unidos. Johnson & Johnson fue dada autorización de uso de emergencia el 4 de febrero y es una sola inyección en lugar de dos. También ahí está la Novavax y la vacuna Sanofi.
So these vaccines are using some new technologies that have previously not been used before. The COVID pandemic has led to this exploration and further development of different types of vaccine platforms. And I say exploration and further development because these platforms were already established some over a decade ago when we had the original SARS epidemic outbreak with MERS, the Middle Eastern Respiratory Syndrome. So those two types of platforms are the mRNA and viral vector platforms. We do have information from previous epidemics like SARS-CoV, which was the first SARS, or even MERS, that showed that these vaccine platforms were both safe and effective. But our research into those vaccines was more limited because those epidemics were very quickly contained.
Nueva tecnología de vacunas. La pandemia de COVID ha llevado a la exploración y mayor desarrollo de diferentes tipos de plataformas de vacunas, como por ejemplo vacunas de ARNM y vacunas vector viral. Estas plataformas han sido evaluadas durante más de una década con seguridad y eficacia demostrada. La investigación se detuvo porque los casos se contuvieron rápidamente. Por ejemplo, los casos de MERS y casos de SARS.
This graphic shows the different types of vaccine candidates and the platforms that they use. So you can see here that the mRNA and the viral vector platforms, those are the newer platforms that we're using for COVID-19. The mRNA includes Pfizer and BioNTech, as well as Moderna. Very similar in their administration. It's 2 shots given either 21 or 28 days apart, but they do require very cold storage temperatures. And that's because mRNA is more fragile. So as it gets warmer, there is an increased chance that it can break down and become ineffective. So we have to keep them stored at very low temperatures. They do remain stable at room temperature for a limited period of time. So that's where we try to schedule as many vaccines as we can. We also have the viral vector type of vaccine. So Oxford, AstraZeneca, as well as Janssen, they are using a viral vector platform. The difference between those is Oxford, AstraZeneca is 2 shots, whereas the Janssen is 1 shot only. We then have 2 vaccine candidates that use already existing technology or platforms, and that would be the recombinant type of vaccine, recombinant vaccine platform. So remember, that's where we take the genetic material from the virus and we put it into a harmless cell, and then it creates protein, and we use that protein as part of the vaccine. So that would be Novavax and Sanofi. Novavax, again, would be 2 shots, and the trial is ongoing. And Sanofi, there is the potential for it to be only 1 shot. And they have had some delays in development, but they are getting back on track. They're hoping that by the end of the year, they'll have another vaccine to add.
Este gráfico muestra los diferentes tipos de vacunas candidatas. Por ejemplo, vacunas de ARNM como Pfizer y Moderna que están siendo utilizadas aquí en los Estados Unidos por el momento. Son 2 inyecciones administradas. Por Pfizer es 21 días de diferencia y para la vacuna de Moderna son 28 días de diferencia. La temperatura del almacenamiento es muy fría porque ARNM es mucho más frágil. También hay vacunas de vectores virales, como la de Oxford AstraZeneca y Janssen. La vacuna de Oxford AstraZeneca son 2 inyecciones con 4 a 12 semanas de diferencia. Está siendo usada en emergencia en el Reino Unido y hay ensayos en curso en los Estados Unidos. Y la vacuna Janssen solo requiere una inyección y se solicitó uso de emergencia en los Estados Unidos en febrero 4. También hay vacunas recombinantes como Novavax y Sanofi. Novavax son 2 inyecciones administradas con 28 días de diferencia y hay ensayos en curso. Y Sanofi tiene potencial para hacer una sola inyección, pero hay retrasos en el desarrollo de la vacuna.
We'll start by discussing the mRNA vaccines. So the 2 vaccines that are currently available use the mRNA technology or nanotechnology. mRNA is an abbreviation for messenger RNA. So it is used to code a message and send that message from one part of the cell to another, or in our case, from a lab to our cells. And that coded message is instructions on how to make a protein. After the protein is made, mRNA dissolves. So it just entirely disappears, and there's really no way to trace it. The mRNA in these vaccines cannot change your DNA. And that's really important. I know that there's been some concern about the potential for that to occur, but that's not how mRNA works or is used inside the cell. When I say nanotechnology, the word nano is referring to the incredibly small size of the delivery capsule. So it's a prefix representing 1 billionth of one, which I've written out here. And technology in the most broad sense is a term that means applied scientific knowledge. So when we put that together, this is very, very tiny applied scientific knowledge.
Las vacunas de ARN mensajero o la ARNM, las 2 vacunas actualmente disponibles utilizan nanotecnología de ARNM. ARNM es una abreviatura de ARN mensajero, que es una forma en que nuestras células envían mensajes para producir proteínas. Una vez que se produce una proteína, el ARNM se disuelve completamente. El ARNM de estas vacunas no pueden cambiar su ADN. Y la palabra nanotecnología se refiere, nano, se refiere al tamaño increíblemente pequeño del ARN mensajero y la cápsula de administración. Nano es un prefijo que representa una 1000.000.000ésima parte de 1. Y tecnología es un término que significa conocimiento científico aplicado.
Because the mRNA is the instructions on how to build a protein, we can actually look at a protein and figure out what were the instructions to make it. And that's the really interesting and exciting part of these mRNA vaccines. In the particular case of COVID-19, we want to give our cells or send our cells the instructions on how to make the spike protein. So that's that red piece that everybody sees when you see a picture of COVID. It's the red spike that sticks out. If we can make that, our immune system can become trained to fight it. By virtue, if we train to fight it off, then we can fight off the infection once we're exposed without ever having to be infected in the first place. And the statement that I have here of vaccines working like a fax machine or a printer and scanner is the exact reason why the COVID vaccines that we have can't cause COVID infection. And I'll go into that with some images.
Las vacunas de ARN mensajero, dado que el ARNM son las instrucciones sobre cómo construir una proteína, podemos leer la proteína y averiguar la secuencia de ARNM que la hizo. En el caso de COVID-19, queremos proporcionar a nuestras células información sobre cómo producir la proteína de pico para que nuestro sistema inmunológico pueda aprender a reconocerla y combatirla. Las vacunas de ARNM funcionan como una máquina de fax o una impresora o escáner.
So this is a sequential image that shows how the mRNA vaccines work. So we start off with this spike protein. And this is a 3D printed model of a spike protein based on microscopy, so looking at it under a microscope and figuring out the pattern. Because we know how these proteins are made and the specific code or information that goes into making this, we can reconstruct that code. And we reconstruct it in the form of this RNA, specifically messenger RNA. What we do with this code that we've worked out from the spike protein is we take the code and we place it into a vaccine, and the vaccine acts like the fax machine. So we have this spike protein, we place it into the fax machine. The fax machine delivers a message from one machine to the next, and that digital message, that electronic message, is similar to this RNA. So we have the protein, goes in the fax machine, we send this RNA. The RNA in the vaccine enters our cells, it's delivered to our cells, and our cells know what to do with mRNA, so they can make a new protein. And that's how the vaccine works. Our own bodies are used, our own cells are used to make this spike protein without ever becoming infected with COVID-19. So we're never exposed to the actual virus, we're only exposed to a small piece that our own bodies have made.
En esta imagen tenemos una descripción de cómo funcionan las vacunas de ARNM de forma similar a una máquina de fax. En esta imagen lo que podemos ver al comienzo es un modelo impreso en 3D de una proteína de pico. Esa información se coloca en la vacuna. En esta imagen lo que es la máquina de fax y se envía a la otra máquina de fax y luego esto se envía a sus células. Sus células usan el ARNM para producir una nueva proteína de pico que le enseña al sistema inmunológico a combatir el COVID, pero nunca se está expuesto al virus, en parte o en su totalidad.
In addition to the mRNA vaccines, we are developing viral vector vaccines. These types of vaccines use other viruses to deliver pieces of COVID-19 to our cells. And the viruses that we use to deliver these pieces are not viruses that can cause infection. So it's really just a delivery mechanism. It's like taking that message that we were sending by fax, and we're making the message a little bit more stable by delivering a either more stable piece, such as DNA, or by delivering an actual protein that has been synthesized in the lab, similar to a recombinant vaccine that uses a virus to deliver that instruction or to deliver that protein that's made. These types of vaccines have been studied since the 1970s, I apologize. They work in a similar manner to the mRNA vaccines, except instead of, like I said, just instructions, they can deliver full protein pieces as well. Once those pieces are delivered, our cells can express it and say to the immune system, let's learn how to fight this off.
Vacunas de vectores virales. Las vacunas de vectores virales utilizan otros virus para administrar trozos de COVID-19 en nuestras células. Los virus utilizados no provocan infección. Este tipo de vacunas se ha estudiado desde 1970, no 1907. Perdón por y funcionan de manera similar a las vacunas de ARNM, excepto que no siempre dan instrucciones. Nuestras células expresan los fragmentos de COVID-19, específicamente la proteína de pico, para desencadenar una respuesta inmune.
So in this image, similar to the previous with the mRNA vaccines, we're showing how the spike protein, once it's made or recovered in the lab, is placed into a virus. And viruses come in different shapes and sizes and forms, but it's placed into the virus that can't cause an infection, but the virus is able to deliver that protein into the cell. And our cell is then either able to express the protein that was placed, or it's able to use the instructions contained in this viral vector to make a new protein.
En esta imagen vamos a hacer una descripción de cómo se utilizan los vectores virales para entregar información. En lugar de usar solo ARNM, los vectores virales pueden entregar otros tipos de información a las células. La información se coloca en un virus inofensivo que entrega el paquete a la célula para su expresión. Es otra forma en que podemos entrenar el sistema inmunológico sin exposición al COVID.
So it's for these reasons between the mRNA vaccine and the viral vector vaccine that the COVID vaccines are unable to produce a COVID infection because they're never directly exposing you to the full COVID virus. They're either sending bits of information or small tiny pieces to produce that immune response, but they're never actually using the full virus.
Por estas razones, la vacuna de COVID-19 no puede producir una infección de COVID-19 porque uno no está expuesto al virus de COVID-19 totalmente.
In terms of COVID-19 vaccine safety, we encourage and ask anyone who experiences a reaction after receiving a vaccine to seek immediate medical care or to call 911. And for anybody who has received a vaccine, a COVID vaccine already, you'll know that they typically ask you to wait anywhere from 15 to 30 minutes after receiving a dose. And the reason for that is we have observed a small number of what we call anaphylactoid reactions, where somebody has a very severe allergic reaction to the vaccine. If anybody has a history of that type of reaction, then we ask them to wait 30 minutes instead of 15. But we want individuals to wait so that we have an opportunity to evaluate for that potential reaction. Following any injection, an allergic reaction typically presents within 5 to 15 minutes. So that's where that 15 to 30 minutes comes in for safety. Additionally, there is a system called the VAERS, the Vaccine Adverse Event Reporting System. This system is a national website, and right from their website, it's a national early warning system to detect possible safety problems in US licensed vaccines. So anybody who receives a vaccine can go to this VAERS website, and if they're having a reaction or they think they're having a reaction to the vaccine, they can input that information and it's tracked nationally. They look at individuals who have received the same vaccine and they try to identify if any patterns are emerging. An additional tool or mechanism is something called vSafe, which is short for vaccine safety. This is actually managed by the CDC and it's available online through any smartphone, so you can use it on the go. And what they do is they track and review your symptoms. After the first vaccine, this is done daily just to check in and see how you're doing. And then after you've received your first vaccine, they check in weekly. They also remind you when to receive your second dose based on the vaccine that you received for your first dose. And if your symptoms become serious enough, or if they're reported as serious enough, someone from the CDC may actually reach out to you to ensure that you're okay and that you're safe, or give further guidance if you need medical care or attention.
La seguridad de la vacuna contra el COVID-19. Cualquiera que experimente una reacción después de recibir una vacuna debe buscar atención médica inmediata o llamar al 911. Usualmente hay un periodo de control de 15 minutos después de recibir todas las dosis de vacuna de COVID-19, o el periodo de control es de 30 minutos si uno tiene antecedentes de anafilaxia o tipo grave de reacción alérgica. También hay sistema de notificación de eventos adversos de vacunas, VAERS, como se indica en su sitio web. Es un sistema nacional de alerta temprana para detectar posibles problemas de seguridad en las vacunas autorizadas en los Estados Unidos. Y también hay vSafe, que es administrado por la CDC y está disponible en línea usando cualquier teléfono inteligente para rastrear y registrar los síntomas de la vacuna. También envía recordatorios para los registros y programación de la segunda dosis por mensaje de texto. Y si los síntomas son lo suficientemente graves, alguien de la CDC se comunicará con usted.
Additional safety points that I want to address include that the current vaccines are considered very safe and are highly effective at preventing COVID-19. And by preventing COVID-19, I mean preventing serious infection. So for Pfizer, it's approximately 95% effective, Moderna 94.5% effective. Now, we do know that there are these variants in the virus that are out there in the world. So the B117, the B1426, different types of variants. And right now we're actually investigating or studying how these variants may impact the efficacy of the vaccine. What we are seeing is, based on preliminary data, these two vaccines, the two mRNA vaccines, do hold up fairly well against these variants. Some others don't perform as well. So that's where we're looking at the broader picture, the larger picture, and we're trying to get the vaccines to where they'll be most effective. What's also nice is, or interesting, is that because these vaccines, the Pfizer and Moderna vaccines, are so effective, I mean, these numbers are the highest that we've ever seen with the vaccine. We're already starting off at such a high place that even if we reduce the efficacy by 10 to 15%, we're still maintaining a 75, 80% effectiveness against severe COVID-19. And that's a huge impact. The vaccines are recommended for anyone over the age of 17. And that's where the Pfizer and Moderna differ. Moderna right now is 18 and older, but there are plans to explore pediatric dosing. These vaccines are also considered safe for women who are pregnant or breastfeeding. What we do recommend or encourage is that if anybody is intending on becoming pregnant, that they receive both doses of the vaccine before becoming pregnant. There are no issues with fertility or infertility that have been identified to date. But we do recommend that only because of the side effect profile, and it's better to have the immunity before you become pregnant. Again, earlier in the presentation, we talked about pregnancy creating a slight immunosuppression effect. So we want to make sure that we are as protected as possible. There's also no evidence to suggest that the antibodies produced by the vaccine or even the vaccine components are passed through breast milk. So it's safe for breastfeeding as well. There's no evidence to suggest that receiving a COVID-19 vaccine is associated with an increased risk of death. So when we look at vaccine safety and we look at the overall numbers, we do see that there are individuals who are in the vaccine trials or who have received a vaccine who have passed away, who have died. But when I say that there's no increased association or increased risk of death, what we look at is we look at the overall effect that the vaccine has, or the outcomes rather, in the vaccine population compared to the general population. So if we see the same rate of occurrence for a particular outcome, then the vaccine really isn't contributing to that. Whereas if we see an increased rate for a particular outcome, then the vaccine would be contributing to that. And we haven't seen that increased contribution. So while yes, unfortunately, people have died after receiving a COVID vaccine, it's not because of the vaccine itself. And if you aren't sure if you should or should not receive the vaccine, please, by all means, speak to your healthcare provider. And you can also go to different websites. So the CDC website contains a lot of really valuable and resourceful information as well.
La seguridad de la vacuna contra el COVID-19. Las vacunas actuales de COVID-19 contra COVID-19, Pfizer y Moderna, se consideran muy seguras y son muy eficaces para prevenir COVID-19. Pfizer tiene 95% de efectividad, mientras Moderna tiene 94.5% de efectividad. Hay una pregunta sobre las variantes del virus. ¿Cómo las variantes podrían afectar la eficacia de estas vacunas? Y según los datos preliminares, estas vacunas se mantienen bien en frente de las variantes. ¿Qué más? Es recomendado para cualquier persona mayor de 17 años. Planes de explorar la dosificación pediátrica en curso. Y se considera seguro para mujeres embarazadas o en periodo de lactancia. Si uno está planeando tener un bebé, es recomendado recibir ambas dosis de vacunas antes de quedar embarazada. No hay evidencia que sugiera que recibir la vacuna contra el COVID-19 esté asociado con mayor riesgo de muerte. Y si tienen preguntas, hable con su proveedor de atención médica si no está seguro si debe recibir la vacuna o no.
just to reiterate the point, the COVID vaccines cannot cause infection. And part of the misconception surrounding that also stems from the fact that many individuals may have received their first COVID vaccine, and then several days later, they report that they are now COVID positive. The tests that we use to look for COVID don't look for the antibodies in all cases. In some cases they do, but in some cases they don't, and especially the nasal swabs, we're looking for the actual virus. If an individual becomes COVID positive, it's not because of the vaccine. The vaccine doesn't influence that type of test. It is possible, however, that an individual is either infected and asymptomatic, or they're in the pre-symptomatic stage when they receive their first injection. So they may have already been exposed, they may already be developing the infection, but they haven't begun to experience any symptoms yet. They received the first vaccine, and then several days later, they have signs and symptoms that are consistent with COVID, and now they have a positive COVID test. It's not the vaccine that caused that. It's because of an actual infection. What is also important to note is that the vaccines do produce an immune response. It can range anywhere from just arm tenderness or soreness to flu-like symptoms, but these symptoms typically resolve within 24 to 48 hours after a vaccine. So if you receive your first dose and you're having these side effects, these after vaccination side effects for 3, 4, or even 5 days, it's more likely that you have an underlying infection and you should be evaluated by a healthcare provider. If you are diagnosed with COVID after receiving your first dose, you can still receive your second dose. We encourage that you wait until you've recovered and no longer required to quarantine, and that you schedule the second dose as close to the target date as possible. Anybody who has recovered from COVID-19 may also receive a vaccine. The only exception to this is individuals who have received monoclonal antibodies for treatment of COVID-19, which is currently available through investigative trial emergency use authorization. Those individuals should wait 90 days, and that's because of the interaction mechanism between the monoclonal antibodies and the vaccine. So we want to make sure that the vaccine is fully effective.
La vacunación e infección. Las vacunas contra COVID-19 no pueden causar infección. Es posible estar infectado o asintomático al recibir su primera dosis, o sea, antes de recibir la vacuna. Si le diagnostican COVID-19 después de su primera dosis, aún puede recibir la segunda dosis. Espere hasta que se haya recuperado y ya no tenga que ponerse en cuarentena. Programe la segunda dosis lo más cerca posible de la fecha de objetivo, y cualquiera que se haya recuperado del COVID-19 puede recibir una vacuna. La única excepción es para las personas que han recibido terapia con anticuerpos monoclonales para tratamiento de COVID-19, que están en ensayos de investigación. Estas personas deberían esperar por lo menos 90 días.
So I want to thank Dr. Marshall Glesby and Dr. Kristen Marks for sharing their slides, the continued efforts of the Weill Cornell HIV Clinical Trials Unit, and all of the clinical participants for their contribution toward changing the world.
Agradecimientos a Dr. Marshall Glesby y a Dr. Kristen Marks por compartir las diapositivas, a la unidad de ensayos clínicos de VIH de Weill Cornell, y gracias a todos los participantes de los ensayos clínicos que han contribuido a la comprensión científica de la pandemia. Su participación individual está salvando innumerables vidas en todo el mundo. Okay. Before we go to questions, antes de comenzar las preguntas, voy a comenzar una encuesta. I'll start a poll, so please fill it out. If possible, this will be anonymous. And I should start now, and you should be able to see a poll on your screen. Questions. This question comes from, so it says, if the mom has a vaccine, any data about the infant gaining immunity through breast milk? Do antibodies traverse into the milk? Si la mamá tiene la vacuna contra COVID-19, ¿algún dato sobre la inmunidad del bebé a través de la leche materna? O si los anticuerpos atraviesan la leche materna?
That's a great question. And while we don't have a lot of data on that yet. We do know that the mother is able to pass antibodies to their infant, and that produces what's called a passive immunity. So it is, in theory, protective. We just have limited information on that.
La respuesta es, todavía no tenemos muchos datos al respecto. Las madres pueden transmitir anticuerpos al bebé y producir inmunidad pasiva, pero los datos que tenemos aún están limitados. And there is a question, actually a comment from Diane Mack.
Yes, I see in the chat.
Yeah.
So I'm reading this comment. So regarding two situations. One is a doctor told a patient that resides in California that New York State had a different version of COVID-19 than California, which is why New Yorkers were hit harder. So for that first point, we do know that there are different variants that are at play in the country right now. We actually know that there is what's considered to be a homegrown variant. So variant B1426 is actually a variant first identified in California, and that's associated with increased virulence, so it's more contagious. The chance or the potential for New York having had a different variant, at least at the beginning of the pandemic, is lower because there wasn't enough time for those variants to begin emerging. New York was initially hit much harder because we didn't know what we were looking at. We didn't know what we were treating or what we were fighting against. So with that limited information, it was harder for us to develop a game plan in response. The second statement is regarding a nurse that stated vaccines are administered according to insurance. The better the insurance, the higher the quality of the vaccine. So that is also untrue. Right now, we have the two vaccines. Both of them have been proven to be almost equally effective, differing by half a percentage point. And oftentimes when it comes to these COVID vaccines, the insurance is not the person paying. These are funded by both state and federal programs. They may ask for insurance information in an attempt to recover some costs, but insurance is not a deciding factor in which vaccine you receive. The COVID vaccine that you receive is dependent upon what is available at that site on that given day.
Hay un comentario. El comentario dice que en el estado de Nueva York preguntan, dice que el estado de Nueva York tiene tiene una variante diferente de COVID-19 que en California. Por eso ha sido golpeado más fuerte, o ha sido más fuerte en Nueva York. Y la respuesta que Jonathan Berry dio fue que en Nueva York se vio más afectada al comienzo porque no sabíamos que estábamos tratando al comienzo. Y la segunda es si una persona tiene mejor seguro, la calidad de la vacuna sería mucho mejor también. Pero esto es falso. Las vacunas son casi igualmente efectivas y el seguro no es un factor decisivo por cuál vacuna uno va a tener. Si es Moderna o Pfizer. El factor decisivo es cuál vacuna está presente. Digamos, si la vacuna que tienen ahora son Pfizer o si la vacuna que tienen en el momento es la vacuna de Moderna.
Another question that is in the chat is, when will we know how long the vaccines will protect us from the illness? Another great question, and the answer is we're not sure yet. We're still investigating the vaccines, not only in terms of the short-term efficacy, but in terms of the long-term protective nature of these vaccines. And we have to remember that the pandemic only started about a year ago. So we're learning in some situations, literally day by day, based on the information that's available. So we can use some models to try and predict or project how much immunity or how long the immunity will last for, but we're also talking about different vaccine mechanisms, so the models may not always be accurate. Right now, the hope is that we'll have at least a period of 2 years for immunity from COVID-19. but again, subject to change based on information that we learn as the days and weeks pass.
Hay una pregunta. ¿Cuándo sabremos cuánto tiempo nos protegerá la vacuna de COVID-19 contra la enfermedad? La respuesta es, aún no estamos seguros. Todavía estamos investigando la vacuna. La pandemia comenzó hace apenas un año y estamos aprendiendo día a día según la información que está disponible. Podemos utilizar o usar algunos modelos para predecir cuánto tiempo durará la inmunidad, pero los modelos no serán precisos, ¿no? La esperanza es que por lo menos tengamos un periodo de inmunidad de 2 años. I think that is all for questions. And yeah, I think that's it. Thank you very much, Jonathan. Thank you, everyone, for joining us today. Muchas gracias, Jonathan. Muchas gracias a todos ustedes por sintonizarnos el día de hoy. And we'll see you next time. Thank you, Jonathan. Have a good one.
Care.
Thank you, everyone. Have a good one, everyone.
Morning, Willamette. Larry here. Good to
Hi,
see
Larry.
you.
You have a question?
Just want to say hi.
Oh, okay.
Hello to Willamette.
Hello.
Jonathan,
hello.
that was a nice presentation. Thank you so much.
My pleasure.
Are you based at Weill?
Yes.
Are you looking at